Good morning, everybody. Myself, Dr. Sejian, Principal Scientist at Indian Council of Agriculture Research, National Institute of Animal Nutrition and Physiology, Bangalore, India. At the outset, let me thank the organizer for providing me this opportunity to share some of my research findings. I'll be talking on the topic climate resilient animals in arid regions, adaptation, emerging concepts and technologies for sustenance. This is going to be my talking points for today. I'll be talking on the climate system and global climate change and consequences, climate change and concept of multiple stresses impacting livestock, advanced thermal indices to quantify heat stress response, advances in assessing the thermotolerance of livestock, skin-based approaches in assessing thermotolerance, goat as the ideal climate resilient animal model, identifying agroecological zone-specific breeds and technologies for sustaining livestock production in the changing climate scenario, genetic approaches to identify climate resilient animals, the conclusion and future perspectives. As always, I like to stimulate the audience by showing some of the pictures which could portray to us the adverse impact of the climate change. This is the European heat wave in 2003, and you can see that almost 35,000 people died for the heat wave. And this is a devastating effect of Indian tsunami in the year 2004, where you can see the gross picture of the devastating effect of the climate change. And this is a Russian heat wave in 2010, where you can see that average increased in the extremely severe heat stress during that particular year. And this is the Thai flood in 2011, where you can see the devastating effect in the thing. And then the East African drought in 2011. In some part of the world, it is drought. In some part of the world, it is flood. And you can see almost the barren land, negligible water for animal consumption. This is the typhoon which killed over 12,000 people in Philippines in 2013. And this is the Uttarakhand flood in India in 2013. Where you can see the devastating effect of the flood, and then the devastating effect of Kerala flood in India in 2018. The entire state of southern Indian state Kerala got washed out. And these are some of the fact about the climate change, and you can see that it was estimated that around 262 million people were found to be affected by the devastating effect of the climate change, and quite alarmingly 98% of such population comes from the developing part of the world. And almost 30 million people are likely to go hunger every year as a result of the climate change and the reduction in the agriculture production. And also World Health Organization predicts like almost 150,000 lives are being consumed for the devastating effect of the climate change. These are the quite alarming data, and that is the reason like the world has united to sign the Paris Summit, the declaration to curtail emissions in each and every country. That's how the climate bill came into the existence. And you can see that global climate change, very important data for us to understand the climate change. You can see that the IPCC projects like around 2.6 to 4.8 degrees centigrade increase by the end of this century. And you can see that I have programmed the average earth temperature from 1860 until 2100. These are the projections given by the IPCC, and this rectangular speaks for the climate variability, wherein from 1860 until 2000, the average global temperature was somewhere between 13.5 to 14.5 degrees centigrade. This is called as a variability. By an evolutionary adaptive mechanism, some amount of year, a few years could be wet year, a dry year, a hot year, or the cold year. That could be the explanation for the variability. But then we are in reality here, and where there is something quite alarmingly happening, especially circulated around 2000. After that, where the industrial revolution have taken its toll, and that's where the world is getting the heat of the average temperature, and the IPCC projects like around 2.6 to 4.8 degrees centigrade by the end of this century, with a central estimate of around 3.7 degrees centigrade. My rigorous experimental finding in the livestock says that even in 1 degree centigrade increase in the upper critical temperature are extremely fatal for livestock survival, and that 3.7 degrees centigrade, which is, in my opinion, is a minimal possible projection given by the IPCC. So it's very important that we need to safeguard our animal population. Moving on to the impact of the climate change on the livestock production, it do not cause the usual suspect heat stress. Climate change causes multiple environmental stresses, especially when the system of rearing of livestock is extensive. which could have direct effect as well as indirect effect. The direct effect by all means the heat stress, and the indirect effect it is like certain disease outbreak, particularly the vector-borne diseases, and less feed and water available as well as low grazing land available. All these could culminate in affecting the livestock production, causing severe economic constraints for the livestock industry. These are the overall impact of the heat stress on the livestock production. See, it could affect the growth, where the body weight, body condition, average daily gain, feed intake, and feed conversion efficiency may get compromised. Milk production, both the quality as well as the quantity of milk can get affected, and lactose, milk fat, and solid non-fat and milk protein content gets reduced. As far as reproduction is concerned, reduced estrus expression, fertilization rate gets compromised, conception rate gets reduced, and follicular genesis and estradiol production gets compromised. And if it is a male, there will be spermatogenesis gets altered. And as far as the meat production is concerned, almost all the major carcass traits and the physicochemical property and other meat quality governing gene expression pattern gets altered. And as far as the disease occurrences are concerned, particularly the vector-borne diseases, trypanosomiasis, bovine viral diarrhea, Rift Valley fever, rinderpest, and theileriosis all have an interlinking with the climate change. But then disease occurrences linking to the livestock climate change is an infancy state, and a lot of research need to be happening across the globe. That warrants more research efforts. And this is the review which speaks about the various impact adaptation of animals to the heat stress. And in collaboration with my international collaborators, I brought out this review. And an important slide from an Indian perspective, you can see that the country produces the largest milk in the world. And you can see some important data this slide has. You can see the distribution pattern in India, the milk production, 25% comes from the crossbred, and 20% comes from the indigenous animal, and 51% from buffalo. Only 4% comes from the goat. And you can see why India, in spite of global climate change, they being a tropical country, they are able to produce more, because the climate change affected reduction in milk production are incurred only from the crossbred animal, whereas the indigenous animal and the indigenous buffalo, wherein you can see that 39 breeds of indigenous cattle and 13 desi breeds of buffalo are contributing to this sustaining milk production, which gives them the status of the world largest milk producer. And you can see the predominant indigenous breeds of the cows, Sahiwal, Gir, Red Sindhi, Tharparkar, Dangi, and Nagori are some of the others, wherein some of the Western countries, including the Brazil, are importing our indigenous germplasm, and then they are going for intensive genetic selection and improving their production efficiency, where in normal Indian condition, when it produces 6 to 8 liters of milk a day, at the intensive selection procedure applied, they produce around 24 to 28 liters of milk per day. That is what the genetic selection can get to do that. Indian government have brought in a lot of policies. For example, milk production in India has increased from 17 million ton in 1950 to 160 million ton in 2015-16. Now currently 206 million metric ton. And National Dairy Development Board has projected that the country's demand for milk would be around 200 million ton in 21-22, which we are comfortably placed achieving at 206 million metric ton, and it is estimated that milk production may decline by over 3 million tons per year due to global warming by 2020, which is already happening. And temperature increase affects the productivity of crossbred cows over the indigenous cows. So this signifies the importance of such indigenous germplasm. Brazil, Australia, and the US are importing indigenous milk animals to develop heat-resistant species. That's what I was explaining. So this gives an importance of conserving the indigenous germplasm, as well as the improving the productive efficiency of the germplasm could be a better way forward, rather than improving the production efficiency of the crossbred cattle, which are going to feel the heat of the climate change. Well, this is a pictorial representation which is another important environmental stress linked with the climate change, that is walking stress. You can see that this is a pictorial representation of two groups of animal, one in the shed and another in the walking. The animal in the arid region usually travel across 7 kilometers in the morning, 7 kilometers in the afternoon for the search of the limited pasture. So we conducted an organized study where you can see that they have been put with indigenous face mask. They have been acclimatized before start of the study to prevent any restraining stress for this animal. The idea is to keep the feed intake intact with the in control animals as well as the walking animals. So they are not allowed to graze during the walking. Only the locomotory activity we have used it to know like how the locomotory activity is having an impact on this animal, and we have established to be having a severe impact on the animal. So effect of walking stress on the growth, physiological adaptability, endocrine responses in malpractice in a semi-arid tropical environment. It's a first of its kind report linking walking stress to be another important environmental stress concerned with the climate change in the livestock production. This is the water stress where climate change impact on the water resources. You can see that in 4 different districts of arid region, Rajasthan in India, where Bikaner, Bhilwara, Jodhpur, and Tonk are the 4 different districts, almost barren land, no water for animal consumption. So this need to be studied. What is the impact of this? And it's still a lot more research need to happen to quantify the water stress impact on the livestock production. Well, if you see in a summer season, these are the stresses that are happening simultaneously: heat stress, nutritional stress, both feed as well as water non-availability, as well as the walking stress. What happens? We are studying only the heat stress effect, but then in a summer season, all these are happening together, particularly when the system of rearing is extensive. This provoked me to develop a concept of multiple stresses impacting livestock production. I can see that this is a pictorial representation of the hypothetical model we have developed, and wherein we conducted several studies and proved the hypothesis. And you can see that if 2 stressors occur simultaneously, the total cost may have severe impact on the biological function. Let me explain the concept. The first part of the picture is a normal happening in an animal, where animal is blessed with some sort of body reserve, and growth, reproduction, milk, meat, and immunity are the productive functions, whereas the basal functions are very vital for animal survival. What happens when the animals are subjected to single stress, for example, heat stress? The animal will not feel the heat, especially in the indigenous animal. They will be able to cope up to the single stress with the help of body reserve. Okay, quite easily. What happens? I made a statement. These are the different type of stresses that are happening simultaneously: heat stress, feed non-available, nutrition stress, water non-available, water stress, and animal have to walk long distance in search of this limited pasture, which can cause walking stress, which are happening in the summer season simultaneously. So therefore, what happens in the extensive system of rearing? When 2 or more stresses are happening simultaneously, the body reserves are not enough to cope up to the stress condition, but then the animal have to compromise the productive function, because they cannot compromise the basal function, which are very vital for survival. So in our experimental finding, we have observed growth is getting first compromised, followed by reproduction, milk production, meat production, as well as the immune status gets compromised. Ultimately, if the stress prolongs, then the animal have to die. This is an hypothetical model, and we proved this hypothetical model through several experimental findings that has led to some of the world's best publication that is emerging from the concept. I'm not here to project that we have published more, but then the concept has been accepted by wide publishers across the globe. Moving on to the second concept, that is advanced thermal indices to quantify heat stress response. You can see that till date, the world is looking for temperature humidity index, and which could be degree centigrade and degree Fahrenheit. And then these are the distribution of various categories of absence of heat stress, moderate heat stress, severe heat stress, and extremely severe heat stress. What happens? Is it the THI, the foolproof indices to quantify the stress response? The answer should be no. Why? It is because it do not take into account other cardinal weather variables such as air temperature, air speed, as well as solar radiation. Wind velocity and the solar radiation, the THI do not take into account. And also with the THI, we cannot predict how many hours per day the animals were subjected to the extremely severe heat stress. To address these constraints here, group of team from University of Queensland, headed by Professor John Gauguin, have established a heat load index, that is using a black globe thermometer reading instead of ambient temperature recording devices, which takes into account both the solar radiation and also the equation as the wind velocity or the wind speed in the equation. And also it addresses the second drawback, that the accumulated heat load index will give you a clue about per day what is the duration with which the animals are subjected to extremely severe heat stress. This is the equation wherein you can see that wind velocity and the ambient temperature recorded using black globe thermometer, which records both temperature as well as the solar radiation. If the cumulative score equals 75, then it is considered extremely severe heat stress, and based on that per day you can calculate where dairy heat load index can give you a clue per day how many hours of duration the animals were subjected to extremely severe heat stress. This is the country-based vulnerability index for livestock production in the projected climate change scenario. I am collaborating with some of the geographers from the Chile, and this is an hierarchical clustering analysis and principal component analysis, which distributes the world countries into 3 clusters. The cluster 3 are safe zone, where predominantly the developed countries are coming from, and the cluster 2, where predominantly the European and some part of the African and Indian subcontinent comes from. Cluster 1 were highly vulnerable. You can see that it is depicted in the red color, where they are highly vulnerable because they don't have any preparatory system in place. And you can see, in fact, India and Sudan are going to be highly affected because they are not falling in the cluster at all. They are separated, not falling in any of the cluster, which gives an alarming situation for these 2 countries in the near future. This shows like which all the country need to be concentrating on developing mitigation strategy. Moving on to the third concept, advances associated with assessing thermotolerance. And these are all the responses we are recording from the animal. That is behavioral adaptation, physiological adaptation, neuroendocrine adaptation, blood biochemical response, metabolic adaptation, cellular adaptation, molecular adaptation, and morphological adaptation. What we are trying to do in this process is that we are trying to identify our program set of biological markers which could be used for future breeding purposes. You can see the coat color difference, almost 4 degrees centigrade between a light and the black coat colored goat. And these are the morphological adaptation, the virtual cattle, the shortest breed recorded on the earth. You can see that the average body adult virtual cattle in comparison to the adult crossbred cattle, and this is what the size. Advantages: they are extremely adapted, they are less feed requirement, and they require less water also. So like what we are trying to do is that we are trying to identify several biological markers using this indigenous germplasm, which could be utilized for the future breeding purposes. This was in collaboration with the Kerala Veterinary and Animal Sciences University for me with my collaborators. And this is the physiological response record, and this is the sweating rate measurement in the scrotum, and these are the physiological response: respiration rate, pulse rate, rectal temperature, skin temperature, and sweating rate measurement. This is the infrared thermometer which is used for recording the surface temperature, and this is the cobalt chloride method for assessing the sweating rate measurement. And this is the rumen temperature measurement. You can see that the bolus gun, where bolus is there, you can see that which works in the RFID principle, which is soaked in the water bath 37 degrees centigrade overnight, and the next morning they have been used and it is inserted into that, so that through RFID principle it can get provide the signal for recording the animal rumen temperature quite every 5 minutes. And these are another technology, that is infrared thermal imaging facility, where we can assess the climate resilience based on surface temperature programming. And this is all the IRT images of different surface temperature in the goat. And you can see predominantly we record in the eye temperature, nostril temperature, and forehead temperature, shoulder temperature, and back temperature and flank temperature, and also the front leg temperature. You can know like the adapted breeds have a very lengthy thin leg to prevent the ground radiation hitting them, so their front leg temperature also would be very vital to record that. And these are the moving on to the next concept, skin based approaches to establish climate resilience. And you can see that these are the novel insight into skin based approaches, because this is the first body part which gets exposed to the skin. Obviously we have segregated them into 2 different biological samples, that is hair, and animals were slaughtered to collect the skin, wherein from the hair characteristics and the cortisol and qPCR from the hair to establish the anatomical, hormonal, and molecular changes. And in the skin, active sweat gland and sweating rate measurement, and histology of the skin, and surface temperature measurement using infrared thermal imager, and metagenomics, transcriptomics, and bisulfate sequencing for studying the skin microbial diversity, and then the differentially expressed genes, as well as the differentially methylated pattern or DNA methylation pattern, which can give a clue about the epigenetic changes. So all these can culminate in assessing the variations among the indigenous breeds, and then decide the thermotolerance which could be identified as the best breed, which could be disseminated to the poor and marginal farmer for ensuring their livelihood security. Moving on to the next concept, goat as the ideal climate resilient animal model. You can see that this is a modeling approach based results by a group of scientists from International Livestock Research Institute, Nairobi, where you can see that the current scenario and the 2050 scenario and 2100 scenario, the poultry and the piggeries are going to be highly vulnerable for the devastating effect of the climate change on the heat stress. Whereas the dairy cattle, beef cattle, and sheep are also getting, but then in every category, if you see by 2050 as well as to 2100, goats are going to be least affected. That is the reason they are projected to be the ideal climate resilient animal model. And this is the latest report globally. This is for the East African region, and this is for globally, where we collaborated with Professor Carvajal from Chile, and we have established that again for the global scenario also, goat is going to be the less vulnerable species for climate change as compared to that of the dairy cattle, pig, poultry, as well as the sheep. That's the reason you can see that predominantly in the tropical countries, the goats are being distributed. You can see that this is a review by our own team where it shows that predominantly the goat population comes from the tropical country. That is because they are better thermotolerant, they have the unique feeding behavior, they are better drought tolerant, and they have the potential to produce less methane and the ability to survive on low pasture and better feed conversion efficiency and high disease resistance as well as low investment for goat farming. So these are the very vital advantages of the goat rearing, which projects them to be which imparts them the capacity to be claimed as the ideal climate resilient animal model. And you can see that behavioral plasticity of the goats. This is the bipedal stance which helps them to browse in the tree, and then they can also have a selective feeding of stem as well as the cactus for meeting their water requirement. And also one cattle can take a shade wherein 6 to 8 goat can easily get compromised in that. So this is another advantage of the goat. So that is the reason this has been projected as the ideal climate resilient animal model, because they could ideally survive on any limited pasture, and their unique feeding behavior provide them that advantage over the other species. And this is what the review we are referring to, that goat as the ideal climate resilient animal model in tropical environment, revisiting advantages over other livestock species. Well, this is the science of the total environment paper, where increasing temperature, importance of the heat stress for cattle farming under future global climate scenario, which gives a clear edge to the goat as the future climate and resilient animal model. Moving on to the next concept, identification of agroecological zone specific breed. You can see that what is the idea now our team is working on. We are trying to screen all the indigenous goat population. We can see in the southern India there are 7 different breeds, and all the 7 breeds we have screened for their climate resilience, taking their behavioral variable, physiological variable, neuroendocrine variable, blood biochemical variable, and then the different molecular chaperone gene as well as growth related gene expression pattern. And then also we are studying their growth as well as the meat production characteristic for understanding their climate resilience, because to claim an animal to be climate resilient, they not only should adapt and also they should produce optimally. And also we are studying their climate resilience based on some novel approaches such as rumen metagenomics and skin metagenomics, and the liver as well as the peripheral blood mononuclear cell transcriptomics approach, as well as the skin based transcriptomics approach, and then the bisulfite sequencing of the skin to know the DNA methylation pattern different to reflect the epigenetic changes that are associated with the livestock adaptation. So based on these variables, set of variables in all these 7 breeds we have completed, and we have established Kodiak breed to be the stand apart breed among these 7 breeds, which not only produces optimally and also survives very efficiently, and also it has the potential to be shifted to different agroecological zones so that we can disseminate it to the poor and marginal farmer in other agroecological zone also. That is a huge advantage. Also equally the Salem black breed and Kanniyadu and Nandidurga are also showing promise, which should be exploited for the future breeding purposes. And so again from this particular concept of agroecological zone specific breed, you can see that how many number of publications have emerged again to show our concept have been accepted cutting across the publishers. Moving on to the next concept, technologies to sustain livestock production. I would just group them into 4 different categories, like housing management, which is nothing but to provide them the appropriate microclimate for their optimum production as well as the survival. And the second set of technologies that are nutritional modification, nutritional intervention to provide the essential micronutrient that are required for their normal survival as well as the production. And also the most important animal genetic approaches, genetics and breeding approaches, which the ultimate goal is to produce more thermotolerant breeds, which could be and then in fact to compare and assess and then identify the best climate resilient species. Within the species, what are the different breeds that are climate resilient, and within the breed, what are the allied animals that are showing excellent climate resilient. Those should be identified for the marker assisted selection or marker identification, so that it could be used for marker assisted selection breeding program to produce more young one. And also most importantly, the disease surveillance measure, wherein these research are in infancy state, and we need to intensify the research efforts, particularly to identify climate change oriented disease occurrences, and that should be linked to the geographical information system, followed by the laboratory research to identify what are those diseases that have a direct linkage, particularly the vector-borne diseases for the climate change, and how to be tackling them. So these are the advantage. And then the microclimate is animal shade structure, shelter design, animal shade, cooling system, and ventilation system. As far as the modification, season specific feeding, fiber feeding, feeding fats and concentrate, vitamins and mineral supplementation, water requirement, livestock diversity, genetic selection, genomic, proteomics, as well as the embryo transfer and stress resistant breed, as well as the monitoring and control of the climate change associated diseases, epidemiological surveillance, rapid investigation of the outbreak, geographical information system should be linked, and then laboratory and field research have to be oriented. These are the different strategies to sustain livestock production in the changing climate scenario. These are the easy to use technology by the farmers, use of the fans, wet gunny bag, and then growing vegetation over the roof and mist cooling. And you can see that automated wetting and drying unit, wherein when the THI exceeds to touch beyond a certain limitations, then automatically there will be for 30 seconds sprinklers will work, and then the fan will be on for half an hour to prevent the moisture, so that the THI remains in the comfortable condition for the animal. This is all are innovative in technologies which could be used to assess the stress as well as ameliorating them. And this is indigenous intravaginal sponges for heat stress induction and synchronization during heat stress response. And then this is costing around 7 rupees 50 paisa, very very cheap, and it works with the sheep, goat, cattle, buffalo, mithun, yak, almost all the species. This is getting worked, and then it is an progesterone impregnated intravaginal device which would be used for synchronizing the estrus. As you know, like during stressful condition, the reproduction gets compromised. When we are using for synchronizing, that is a huge advantage to maintain their reproductive efficiency. Lighting management to improve cattle, and replacing the incandescent bulb with the luminous bulb is the Bacillum luminous, which is a company based in Germany. More yield through biologically effective light. And then the more moving on to the next concept, genomics approach to identify climate resilient. What our team currently focusing on to identify more biological marker. To do so, what is that our target? We are targeting rumen microbes as well as the skin, and then the sweat gland. What we are trying to do in this process, epigenetic changes in the skin. For example, bisulfite sequencing of the skin will give me an indication about the differences in the DNA methylation pattern, and histone molecule modification, non-coding RNA, as well as the chromatin remodeling, which will give on generation interval adaptive changes. Whereas from the skin, what we can understand, genetic traits in skin and liver governing climate resilience, sweat metabolomics, skin metabolomics, skin metagenomics, skin and liver transcriptomics. All these can give a concrete adaptive, unique adaptive potential of the animal. And then the rumen microbial diversity, particularly where rumen metagenomics, microbial diversity can be established, and quiet and circulated genes and genomes pathways could be analyzed, so that we can identify more biological marker which can give an indication about novel approach to establish climate resilience based on rumen microbiome associated adaptive potential. All these could help to identify the climate resilient breed in the goat. And moving on to the next concept, rumen microbiome associated adaptive potential assessment. And you can see that this is an experiment wherein we compared the rumen metagenomics with the 3 different breeds, and you can see that at different taxonomic level, what are the real difference that are happening as a result of the subjecting these breeds to the heat stress. In the Ostner body, 1 phyla, 6 class, 2 genus. In Malabari breed, 2 phyla, 7 class, and 4 genus. Whereas in the Salem black, only 1 phyla and 2 class and 1 genus got affected. What what is the inference we could take from this is that there are some sort of responses that is happening when these breeds were subjected to the heat stress, and Salem black breed as compared to that of the Osmanabadi and Malabari breed have maintained their rumen microbial composition intact, which shows their which could be related to their extreme climate resilience, which means like could be a novel approach in the future to this could pave a way forward approach for assessing the thermotolerance based on the rumen microbial diversity. That is what it is, novel insights into rumen microbe based adaptive potential establishment in the goat. And moving on to the next concept, old transcriptome associated adaptive potential, and you can see that the differentially expressed genes you can see in 3 different breed, and in the Salem black breed it is very less as compared to that of the Osmanabadi and more in case of the Malabari breed. And in terms of the exact number, in Osmanabadi 326 genes got upregulated, whereas 279 genes got downregulated, whereas in Malabari 687 genes got upregulated and 635 genes got downregulated. Whereas in the Salem black breed, it is 314 genes which got upregulated and only 196 genes got downregulated. This means, again, from the transcriptomics perspective, Salem black breed maintain its potential, so that there are not much differentially expressed genes as comparison to that of the other breed, which gives them the edge over the other breed for their climate resilience capacity. Moving on to the identification of biomarkers for quantifying heat stress response in livestock. These are the set of variables we have identified to be the markers. For example, in the lymph node TLR1, 4, and 5, there is nothing but toll-like receptors, and thyroid gland thyroid hormone receptor, respiratory tract respiration rate, and hair slick hair gene, and in the blood hemoglobin, packed cell volume, cortisol, triiodothyronine, thyroxine, and then each protein and each factor 1, interleukin, and NRIM1. And then the skeletal muscle, superoxide dismutase and nitrous oxide synthetase. In the liver, there are several enzymes that are established to be the ideal biomarker for quantifying heat stress response. From the skin, it is prolactin receptor gene and fibroblast growth factor. These are all the things which we have identified to be the ideal biological marker. And this is the invited review from the small ruminant research, wherein we have covered almost all the biological marker genes for resilience to heat stress in small ruminants review. And this is another review, prediction models, assessment methodologies, and technological tool to quantify heat stress response in ruminant livestock. Again, it's an important review which can give you all the advanced methodology. Moving on to the next concept, epigenetic changes studies to identify climate resilient in animal, and nothing but bisulfite sequencing to assess DNA methylation pattern difference. And you can see the differentially methylated regions, and you can see in the Kanni, it is only we have done in two among the 7 breed, we have identified the best 2 breed and we compare them because it's a very costly procedure. You can see that Kanni Adu and the Kodi Adu, the hypomethylated and hypermethylated are significantly higher in the Kanni Adu as compared to that of the Kodi Adu breed, whereas the differentially methylated genes are also significantly lower in the Kodi Adu breed as compared to the Kanni Adu breed, indicating the better epigenetic changes reflected by the Kodi Adu breed, which has stabilized epigenetic changes associated with their thermotolerance as compared to that of the Kanni Adu breed, both at the differentially methylated region as well as the differentially methylated genes. And these are the pathways for the KEG pathway, stress pathway at the CPG region in the genome, and you can see that both the breeds mostly it is metabolic activity controlling pathway, whereas in the Kanni Adu breed typically thyroid hormone signaling pathway and metabolic pathway, whereas in Kodi Adu breed typically it is carbohydrate digestion and absorption, vascular smooth muscle contraction, and protein digestion and absorption pathway, which could give a clue about how they are maintaining the metabolic activity related heat production and how they consume the feed for the production to assess their climate resilience. Moving on to the last concept, that is integrated farming for doubling the farmer income, and you can see that this is an innovative integrated model with the goat, rabbit, poultry, and fodder production, wherein it is a collaborative effort, and the individual units are looks like this: the azola tank, the layer unit of the poultry, and grower unit of the poultry, and rabbit unit, and goat unit. 6 goats can be reared, as well as the fodder production or the hydroponics on the roof. So this is what could be used for the farmer, so that even if one enterprise is failed, the advantage of integrated farming is that like the other enterprise can come handy for the poor and marginally marginal farmer, especially to ensure their livelihood security. So that sets the tone for creating such type of integrated designs for the farmer to take an advantage of that. I would like to conclude my talk by saying that research aiming at improving livestock production under changing climate scenarios should aim at countering multiple abiotic stress simultaneously rather than single stress, and efforts are needed to identify more indigenous agroecological zone specific breed which have the ability to survive in different agroecological zones so that they could be disseminated to the poor and marginal farmer. Sustaining livestock production in the changing climate scenario must target on improving the production efficiency through strategical intervention to augment livestock production, and also metagenomics and transcriptomics approaches are providing scope for identifying better climate resilient livestock accurately, and bisulfite sequencing NGS and next generation sequencing analysis helps to identify epigenetic changes associated with the thermotolerance in the goats. And these are the future perspective livestock vision for 2050. If you can see that climate resilience, how we can achieve this, and you can see that this is the exploring the unconventional feed resources, and then the predictive livestock early warning systems, and then exploring the genetic potential of the indigenous breeds, and then improved balanced animal nutrition and balanced diet, identification of ecological zone specific breed, vaccination technology including developing vaccine for rumen methanogens, nanotechnology in animal production, climate smart animal agriculture, and bridging the knowledge gap between the climate change and livestock diseases. And as I already told, that disease linking with the climate change, very less reports are available, and we have to bridge those gap now. And innovation in the methane reduction using, for example, quantifying using nose rings. So that type of innovation, especially the green feed, is upcoming technology that should be used to quantify the methane accurately. Advanced thermal indices, more refined thermal indices need to be developed, and water resource management and genetic and the breeding managemental strategies, genetic selection, and then interrelationship, establishing the interrelationship between soil, climate, and livestock, and to propagate goat as the ideal climate resilient animal model, and applications of the advanced molecular biological tool, for example, NGS tool to assess the climate resilience capacity of the indigenous animal, low carbon livestock development, precision livestock farming, and artificial meat utilization, and climate smart livestock with the farmer centric approach of integrated farming are the way forward approach. These are the future research issue. We are propagating all till date. We have only concentrated on the production trait for the breeding policy. Now it's time to revisit, and then it should be an amalgamation of production trait, adaptation trait, and low methane emission trait per unit of feed consumed. This is for the new breeding policy we are propagating. Well, it's a proud moment to share with all the audience over there the new Center for Climate Resilient Animal Adaptation Study at the ICAR, National Institute of Animal Nutrition Physiology. In my institute, I have established center which has the state-of-the-art climate chamber facility. I told state of the art because it has all the drawbacks of the world climate chambers address, and it's one of the best facility where animal can communicate with each other with a travis, so that they are socializing animals, so to prevent socializing stress. So this has been taken into account, and also the floor space is calculated in such a way they have a very free movement, so that it prevents a restraining stress. And also their individual urine and fecal collection trays are there, and we also have all the setup for quantifying the stress response. And to my left is the thermoneutral zone chamber, where we can induce the thermoneutrality of 24 to 26 degrees centigrade very easily, because we are going to identify more biological marker, we know more refinement. So this is the uniqueness of our climate chamber, where we have a thermoneutral zone chamber, because in the shed, especially when you keep even with the cooling devices, the temperature may not be coming down below 34 degrees centigrade, whereas in this chamber it is possible to induce the appropriate thermoneutral zone of 24 to 26 degrees centigrade. And this is the right side. The chamber has a 12 holding capacity of a small ruminant, and there is a provision for altering the temperature from -5 to 55 degrees centigrade, which means both cold stress as well as heat stress experiments are pretty much possible. This is a proud moment again. 4 Springer books I have published in collaboration with world best reputed researcher for environmental stress and amelioration in livestock, and climate change impact on livestock adaptation and mitigation, sheep production adapting to climate change, and climate change and livestock production, recent advances and future perspectives, which will give you some of the latest technology of non-invasive methodologies also. So these are the way forward. I am disseminating more knowledge through this book, and as a request from the publisher, I need to propagate it in any scientific program. The interested audience could make use of these all important sources of information available for sustaining livestock production in the changing climate scenario. Special thanks, the acknowledgement to the organizers for providing me this opportunity, and also my acknowledgement are due to all my international collaborators and my current institute directors, and then my sister institute collaborators, and most importantly my students. They worked hard with me to generate this data, wherein we can develop more information which are the hidden intricacies as far as assessing the climate resilience into consideration. So they are the team member which works, and I'm representing the team, and especially my students and Dr. Shilpa, who are elaborately working on metagenomics, transcriptomics, and epigenetic changes study. She's been instrumental, and currently she's pursuing a PhD at the Justice Liebig University at Gießen, Germany. So contribution is very, very immense as far as these data generation is concerned. Thank you very much for the opportunity. I will be prepared to take up some question. My email ID and my mobile number has been provided to you. Feel free to contact me for any sort of queries or any sort of future collaboration that could be taken up, and I'll be more prepared to answer you, even if it is the simplest of the query. I can be prepared to answer you guys, and then also we can also establish a very good networking for future collaboration as well. Thank you very much.